Now we'll cover some of the other gram-negative bacilli, coccobacilli that we've not previously covered um, in the previous modules. And so these organisms typically do not grow on the conky, um, and they also usually require some sort of special media to help them grow to present their characteristics as well. Your Vibrio species are associated with brackish water or salt water. And of course, you would get this through exposure of ingesting that water or um, through exposure through seafood, especially if it was undercooked seafood. So these organisms present with a gram-negative curved or comma-shaped type of gram stain. And so you can see a few of these organisms that are kind of rounded here. This is a good example here where it's kind of got a little bit of a curved shape to it or a comma type shape. And this looks like this was probably a culture done off of a blood culture bottle or a gram stain performed on a blood culture bottle. So the morphology would be a medium to large opaque iridescent type of organism or colony. And so you can see down here at the bottom where they kind of have that opaque look. And so sometimes like something that's opaque, when you kind of tilt it, you can kind of see a little bit of the colors in it, almost like an opal. So it might have a greenish type hue to it. They can be lactose or non-lactose fermenters. They are motile. They do have a single flagellum. Oxidase positive, and they do ferment glucose. With your Vibrio infections, there are a couple of different types of species that have been implemented in different types of infections. So for example, ear infections, wound infections, gastroenteritis, and then you can also have Vibrio parahemolyticus, which causes an gastrointestinal type infection through consumption of seafood. So you can see a picture here of that seafood. And of course this would be a type of a skin infection here. Vibrio cholera, this does produce cholera, which would be a profuse watery diarrhea that can cause dehydration. This is also known as rice water stools, and it can also cause hypotension. This is a non-epidemic diarrhea and extraintestinal infection that has been associated with a particular strand of this Vibrio. And there are 3 major groups, and they're all kind of divided out. They have different labels to them, but there are some different types of strands of the Vibrio cholera. And so what they do is they produce a cholera type toxin, and so it causes those mucosal cells within the GI tract to hypersecrete water, and that's where you get that watery diarrhea or that rice water diarrhea. And of course they have different enterotoxins that are associated with these different strands. Aeromonas are inhabitants of aquatic environments throughout the world. And of course these too can be ingested through contaminated food or exposure to contaminated water and soil. And then they can also be introduced through some sort of traumatic event like exposure from a fish fin or hooks. And so with these infections, they do present with a gastroenteritis type infection due to the various toxins that it produces. And of course it too can cause the hemolytic uremia syndrome and kidney disease. So they can be identified based on several characteristics. With these organisms on the gram stain, they do appear as straight rods with curved ends. So you can see here where these gram-negative organisms look a little bit more rounded or plumped on the ends. And so that's kind of characteristic of this particular organism. Their morphology would appear as large, round, raised, opaque colonies on blood agar. They can be lactose or non-lactose fermenters. Oxidase positive, they ferment glucose, and again they are also motile. Campylobacter jejuni is found within the gastrointestinal tract of animals, especially poultry. And so it's transmitted through direct contact of handling of the infected pets or animals, or through indirect contact through consumption of anything that's contaminated with this organism, like water, dairy products, or improperly cooked poultry. There have been reports of person-to-person or sexually transmitted infections. And so the Campylobacter species causes gastrointestinal or extraintestinal infections. So with your extraintestinal infections, this will happen with patients that are immunocompromised. So that this would be an infection outside of the intestinal tract. Your gastrointestinal infections is self-limiting and does not require antibiotics. However, you can have post-infection complications, and an example of this would be the Guillain-Barré syndrome, which causes the neurological complete shutdown of the body. The patient ends up going on life supports when they have this syndrome. Campylobacter jejuni is a small, curved, gram-negative rod, and it can stain very faintly, but it also has a characteristic seagull wing type appearance. So if you look here at the bottom, I'll see if I can't point out some, this one right here. So it kind of has maybe like a little V shape to it, but you can see where it just looks like a bird kind of flying with its wings spread out a little bit. It is very fastidious, it is a slow grower, and it requires a microaerophilic environment. So there are, when they culture this in the lab, there are special bags that they will put these plates in when they are suspecting a Campylobacter infection. And so these bags will help to produce this particular environment. They are cultured at 42 degrees Celsius for optimum growth. On blood agar, they appear as non-hemolytic flat gray mucoid colonies. And if it was plated on the special plate, these plates are called the Campy plate or Campy blood agar plate. And so what these agars do is actually allow for the organism to grow by itself due to the different antibiotics and stuff that are in there. Then it inhibits the growth of the other normal flora that you would find in the GI tract, because obviously if you're expecting this in the GI tract, you would be doing a stool culture, and so with a stool culture you're going to have a lot of normal flora that would grow. So like your E. coli and Pseudomonas, then you would want to inhibit those growths so that they do not overpower the Campy. These organisms are oxidase positive, catalase positive, and hypurate hydrolysis positive. They have a very darting motility when you look at these on a wet prep. So these organisms will, they're very active when you look at them on a wet prep, and I do believe that there is a video of that in your lab session. The direct detection would be used for these as well, so something like PCR or the automated testing for the nucleic acid testing. Helicobacter, a species, or H. pylori, Helicobacter pylori. These organisms colonize the mucosal layer of the stomach, and they will actually live within that gastric mucosal layer. If you have an infection with the H. pylori, you will get gastritis, and it can also cause the peptic ulcers, and it can also progress to a gastric cancer. These organisms do produce urease, which will cause an increase in the pH. And so what happens is it will actually convert the urea to the ammonia and bicarbonate, and this will actually neutralize the stomach acid. And of course, you know, your stomach acid helps to break down things and prevent any sort of growth for anything that's not supposed to be there. and then the ammonia within the stomach will actually start to become toxic and break down those epithelial cells within that gastric lining, and then that's where you start getting the irritation and the peptic ulcers will start to form. It produces the Kag protein, which will help to increase the risk of those peptic ulcers. And so these organisms are microaerophilic, so again, they require a special environment for growth. They are curved gram-negative rods, and they can have a spiral shape to them. Oxidase positive, catalase positive, and a rapid urease positive. So if you were to do a rapid urease test, or even a non-rapid urease test where you actually inoculate the broth, or you would inoculate the agar slants, then those would be positive as well. This organism is usually not cultured because there are more rapid ways to identify this organism. Some of those ways to detect it would be to include a gastric tissue biopsy sample, and you could stain that sample where you're actually looking for the organism within that stain. And then you can also take that biopsy material and put it into a urease broth or some sort of rapid urease detection device, and those rapid kits can usually give you a result within a few minutes to a couple of hours. And so this kind of helps give you a presumptive identification by using these rapid tests. You can also do different automation tests, enzyme immunoassay, where you're looking for the antigen in the stool sample. You can also do a urea breath test, although I don't think that most places are using these anymore because there are other rapid methods. But if for any reason you couldn't do a biopsy sample or stool sample, then there is the urea breath test, where it actually is detecting the amount of bicarbonate that's going to be labeled within that patient's breath. And then you also, if the sample was sent for culture, then it would be cultured on chocolate agar. It could be used for brucella, or even like the Thea-Martin agar. So these agars could be used to help culture this organism if a culture was needed for any reason.